Thanks. Hi, everybody. I'm Chloe. I just like to say thank you so much for inviting me here to speak today. It's my first time ever in Thailand, and it's an absolute privilege to be able to be here to present my work and also hear all the other amazing work that everyone's doing. So thank you. So I'm from the University of Kent in the United Kingdom, in Alessia Buscaino's lab, and I'm going to present a mixture of work I did during my PhD and now a project I'm working on as a postdoc. So for my project, I've been looking at exploiting intra-species diversity for a range of different biotech applications, and my focus has been on biofuel and food. So in the lab, we work with fungi, and these are categorized into two main groups based on their morphology. We have the yeast, which are characterized by budding morphology or fission, and then we have the filamentous fungi, which are also referred to as molds, and these are characterized by their filaments known as hyphae. And these hyphae can form very branched networks called mycelium. There are also some yeasts that can switch from yeast to filamentous forms, and these are referred to as dimorphic, and this switch is usually under stressful conditions. When people tend to think of fungi, they sometimes associate them with negative things, whether this is an infection or it could be mold on bread, for example. But hopefully I can demonstrate to you the great potential behind fungi and their uses in biotech. So first I'm going to talk about my work looking at diversity within yeast and their role for biofuel production. So during my PhD, I worked with a yeast called Saccharomyces stipidus. This is a non-conventional yeast that's found on decaying wood or in the gut of beetles that inhabit this decaying wood. And this yeast is quite unique in the sense that it has the metabolic machinery to metabolize and ferment pentose sugars. It's also known that stipidus has the highest native capacity for xylose fermentation of any known microorganism, and it was for this reason that we were interested in its application for biofuel production. So biofuels are characterized based on the biomass that they're produced from. So we have first, second, and third generation. But I was particularly interested in second generation biofuels, and these are produced from lignocellulosic biomass. This is a broad term to describe agricultural waste, and we're using non-food sources of biomass. So examples of this could be things such as wood chippings or food waste, for example. So the exact composition of the biomass varies depending on the exact type that you use. However, as you can imagine, the structure is quite rigid and complex, and therefore pre-treatment is required to break open the biomass and release the sugars within it. Within these sugars, there are a mixture of pentoses and hexoses, and these can then be fermented using microorganisms such as yeast, which produce ethanol for biofuel. It's crucial that the microorganism chosen for fermentation can ferment both types of sugars for the process to be as cost efficient as possible. So when we think of using yeast in fermentation, the conventional yeast Saccharomyces cerevisiae is normally a top candidate. This is because it's a very well established fermenting yeast already used in industry. However, cerevisiae cannot naturally ferment pentoses, and despite genetic modification to achieve this, transport of the pentoses into the cells remains a rate limiting step. So it's for this reason that we were interested in stipidus, as it can naturally ferment both pentoses and hexoses without needing engineering. So during my PhD, I worked with a collection of different stipidus natural isolates sourced from two different yeast database collections, and these were sourced from various different habitats, different countries. And then we collaborated with a company called Singer Instruments, and this company design and build robots to automate and accelerate scientific research. So we wanted to see if we could come up with a way of screening this strain collection in a range of industrially relevant conditions to see if some natural isolates were better suited than others. So the first robot I got to use is this one here called Pixel, and this was used to transfer liquid cultures of the strains onto agar plates into two different array formats. We had the 96 array, where each strain was replicated just once, and these were used to observe the growth trends studied. And then we had the 384 arrays, where each strain was replicated around 8 times, and they were distributed in a random order on the plane. Once these were created, they could then be replicated onto the different industry relevant conditions tested, and this was done using the robot called Rotor here. So this was a big stamp, and it would come and transfer from one plate to the next. So it's a very quick method of screening a large number of conditions with a large number of strains. So here I'm showing an example of the ethanol screening. It's probably quite hard to see because the screen's a bit small, but we tested a range of different ethanol concentrations, for example, and although you might not be able to see it, some strains were able to tolerate high ethanol concentrations compared to others. So this screening was quantified for all of the conditions tested to rank the strains for the overall fitness for biofuel production. And what was quite nice to see was that there were actually a few stipidus strains that ranked higher than Cerevisiae, demonstrating the potential of using this yeast for second generation biofuel production. Not only did we see differences in their growth performance, but we could also see differences in their phenotype at a single colony level from the data observed. So here I'm showing the reference strain of Stipidus. The genome of this strain was sequenced and published, so therefore it was used as a reference throughout my work. And as you can see, it had a round, smooth colony, which translated into a single budding cell morphology, which is expected for this yeast species. However, I decided to pick a natural isolate here that you can see has a wrinkly colony morphology, and this translated into a filamentous single cell morphology. And this had never been reported before for this yeast, so I was very interested to try and determine why and how it could be beneficial. Due to this filamentous morphology, the strain exhibited biofilm forming phenotypes. So this assay here was measuring sedimentation, which is the settling of cells in liquid culture. And not surprisingly, as there were a large number of filamentous cells, this strain sedimented much faster than the reference strain. And this is a beneficial phenotype to have downstream processing in ethanol production, as it makes it easier to extract the ethanol from the cells. We are a genomics lab, so we wanted to see whether there was a genetic reason behind the phenotype we were seeing. So a range of different techniques were used to study this. The first technique on the left is Chef electrophoresis. This is a technique used to study the karyotype. So each band on this gel represents a chromosome. We obviously knew the chromosome sizes for the reference strain, and we could see that there are a few slight differences based on the separation profiles on the gel. So then we decided to sequence the genome of this strain, and here I'm just showing the nanopore sequencing, which was used to look for large-scale rearrangements. So this circular plot here is a line in the genome of the reference strain to the strain that I chose to study. There were no large-scale rearrangements, but it did correlate the differences in chromosome sizes we'd seen on the Chef gel. I'm not going to go into too much detail for it, but a reason for the different chromosome sizes was due to a difference in the number of repeats. For example, this data here is comparing the number of repeats within the centromeres. So Stipidus contains regional centromeres marked by retrotransposons, and as you can see, for example, in centromere 4, they differed significantly in their number of repeats within the centromeres, and then I was able to use CRISPR-Cas9 to tag a centromeric protein and observe how this difference in centromere structure affected their function and how this could relate to the phenotype observed. So through this study, we'd seen that the genome of Stipidus is plastic, and this plasticity was mainly due to a difference in the number of repeats within the genome of Stipidus. And this is a quite unique trait in the sense that we can utilize natural genomic and phenotypic plasticity to find strains that are naturally better suited for a particular application. So I want to apply this sort of screening to the next project that I'm working on in the lab. So now I'm going to talk a bit about my work on filamentous fungi and how they can be used for food production. So I'm sure you're all aware, but fungi are used in many different food and food products, as well as eating the fungi themselves. This new project that I'm working on is part of a consortium known as the Microbial Food Hub. This is co-led by Imperial College London and a large number of different universities in the UK, as well as a number of different industrial partners. The aim of the Microbial Food Hub is to establish alternative protein sources, and these are categorized into three main fields based on how they're produced. We have the fermentation-based, the plant-based, and the cultivated. The main need for these alternative protein foods is that, as a whole, the food that we tend to eat has a massive environmental footprint. This is mainly due to the fact that we eat a lot of animal-based products. So as you can imagine, the environmental footprint of this is massive. So there is a need to find novel foods that are going to have a low environmental impact, but are also going to be nutritious and tasty. So this is where the Microbial Food Hub comes in. The different partners focus on different parts of alternative proteins, but at Kent, we are focusing on biomass fermentation. So this refers to growing fungal cells with a high nutritional content. And I'm not sure if you'll be aware of this company, but the best well-known one in the UK of this sort of project is Quorn Mycoprotein. This company use massive scale fermenters to ferment filamentous fungi that is high in protein and essential amino acids. For this, Quorn use one filamentous fungi, Fusarium phenomatum, and this fungi was originally isolated from soil in the UK, and it has GRAS status, which means it's generally regarded as safe for human consumption. They use submerged fermentation, and they've obviously optimized the reaction conditions to generate the highest amount of mycoprotein in a short amount of time and be cost effective. However, there are a number of limitations of this fungi and how Quorn currently produce their mycoprotein. For example, they use one natural isolate of Fusarium phenomatum, and there's been no strain development since the 1970s when they first started using this. In addition, they rely on a single feedstock, which is using wheat-derived sugars. Obviously, these are expensive as they require a lot of processing, and the use of this feedstock for food is not ideal. So this is where the project that I'm working on now comes in, where, as I mentioned before, we're going to try and exploit natural genomic and phenotypic diversity, but this time in filamentous fungi, to try and see if we can find filamentous fungi that can utilize waste feedstocks to produce mycoprotein instead of the wheat-derived sugars. So this project is split into different parts. I've only started the project a month or two ago, so it's very early on. But the first part is to try and find waste feedstocks that we can grow the filamentous fungi on. To do this, we need to have a reliable year-round supply of this feedstock for the process to be scalable. We also need to have little variation between batches. So if we're getting a waste from summer to winter, we don't want too much variation. We want to have minimal pre-processing to keep the costs down. And we want the waste feedstock to have a high nutritional content for the filamentous fungi to grow on. So to do this, we've set up a number of collaborations with local companies to try and use their waste feedstocks to grow the filamentous fungi on. And the term waste feedstock is not the best term to use because the feedstocks we're using are not necessarily waste. They're just currently discarded, but they still have a lot of potential, and that's what we're going to test. So the first one is with a local company that makes cider, and they have many apple orchards, and they harvest the apples once a year. The apples then go through a process where they're squished, the apple juice is taken away, and everything else is discarded normally. So we're trying to see if we can utilize that apple pulp to grow the filamentous fungi on. The next is with a distillery, and obviously one of their feedstocks is grain, and we get left behind the spent grain. And again, that would traditionally just be discarded, but again, we're trying to see if we can utilize this as a feedstock. The next is with a company in the UK that makes sandwiches. They're one of the largest UK sandwich makers. And I don't know if it's the same here, but if you buy a sandwich, you would not expect to have the crust. And that's because they are always thrown away. So you can imagine the amount of waste that is generated from that type of production. And the last type of waste that we're trying to see, well, it's not a waste, but we're trying to see if we can utilize seaweed to grow the filamentous fungi on. And this is in collaboration with a company called Nutrisan, and we're using Yorva species. And the nutritional profile of seaweed is quite unique compared to the others. So I'm really looking forward to seeing how the filamentous fungi will be able to potentially utilize this. So we have only just recently received these different types of waste, and we are still trying to figure out how to process it for it to turn into something to grow the filamentous fungi on. This is an example of us trying to turn the bread and apple pulp into some sort of media to grow yeast on. We tested Stipidus and Cerephecii. We wanted to see if yeast could use it first before going to filamentous fungi. Obviously, these two yeasts differ in their metabolic pathways. Obviously, Stipidus is a bit more diverse compared to Cerephecii, and the processing of these wastes was very minimal. We just took 10 grams of each and added water and autoclaved it, and not surprisingly, both types of yeast could grow on it, which was really cool to see. So not much work is needed to find a feedstock for the fungi to grow on. This was also the same for one filamentous fungi, Rhizopus oligosporus. This species did not necessarily really like the apple pulp, but it did like the bread ends. And this was just a very initial screening. So a lot more work is needed to be done, but hopefully I can demonstrate that these wastes can actually be used to grow fungi on. The next part of the project, once we've figured out a way to turn our waste into some sort of media to grow the fungi on, would be to evaluate phenotypic and genomic diversity in the filamentous fungi to see if we can find the strain that can utilize the most types of waste feedstock and generate the highest amount of mycoprotein. To do this, I'm in the process of building up a strain collection of filamentous fungi, as we currently did not have many in the lab. I decided to focus on 6 different species with the aim to have a large number of natural isolates within each. The criteria for these filamentous fungi was that they all needed to have GRAS status, be safety level 1, be non-pathogenic and filamentous. We have already started to get some strains from the ARS culture collection, but we are happy to potentially explore other opportunities for this. The next part would be to try and figure out a way to quantify the growth of these filamentous fungi. I don't know if many of you have worked with filamentous fungi before, but they are slightly more challenging compared to yeast. For example, yeast growth can be easily quantified by measuring OD, but this cannot be applied to filamentous fungi. I don't know if you can see it, but when grown in liquid culture, they form big masses. So obviously OD cannot be measured. So I tested a biomass staining method. So this relies on the biomass settling in a plate, which can then be stained using a dye such as crystal violet, and then this can then be quantified using a spec machine. And the initial testing of this seems to work pretty well. Not only does it allow for a large number of repeats, but it's pretty quick, and we're going to be working with a large number of strains and different conditions. So it's a good way to have a high throughput screening method initially. Once we found the strain that can utilize the most types of waste feedstock, we could then perform measurements such as dry weight to have a more accurate idea of growth rates. Not only will we be looking to see how they're growing, but we'd also be interested to see their morphology, whether this is them on an agar plate or the spores or mycelium that they produce, because I think it's very important to be sure that these sort of traits, they're obviously going to influence the texture of the mycoprotein that's going to be produced. Hopefully, with the large number of strains that we're going to be testing, we hope to see some phenotypic diversity. We will then want to see if there is a genetic basis behind this. So we will then be using techniques such as whole genome sequencing to compare their genomes to one another. The last part of the project would be to evaluate the nutritional profile of the mycoprotein produced and to be sure that it's going to be safe for human consumption. So we first need to figure out a way to convert the fungal biomass, which doesn't particularly look very appetizing at first, into something that people will actually want to eat. And then we will need volunteers to obviously want to try and taste it and let us know what it tastes like. But this will obviously be done after we know that it's safe. So we'll be testing for mycotoxins as well as comparing the nutritional profile. We want to be sure that it's going to be high in proteins, low in fat, for example. And I think it's also important to perform a life cycle assessment to be sure that we're aware of the environmental footprint of the mycoprotein that we're producing very early on. So to conclude, I'm sure you're all aware, but there is a huge potential of fungi for numerous biotech applications. And if you can utilize genomic and phenotypic diversity within a species, it eliminates the need for strain engineering. And if we can utilize waste feedstocks, it provides a far more sustainable way to produce many different compounds in a much cheaper way. So I'd just like to thank everyone that's helped during the PhD and the collaborators now on this project. And thank you all for listening. Thank you.
So thank you, Dr. Croy. Yes. So now we have around 10 minutes for Q&A. So if you want to ask questions, feel free to raise your hand and the microphone will go to you. Yes. Thank you for your talk. I would like to ask about what are the advantages of using filamentous fungi as protein compared with a well-known Saccharomyces cerevisiae for a single cell protein?
One of the main things with the filamentous fungi, the mycoprotein, is obviously the morphology of the filamentous fungi. Their filaments provide a texture that resembles meat-like products. So that's one of the main reasons. They have a much higher protein profile compared to yeasts. So I'd say they're the main reasons. From the brief work that I've been working with them, they obviously, I think they grow much faster compared to yeast as well. So I think they're probably the main advantages.
Hi, thank you for a great talk. I was really fascinated by your work on the Cephasomyces stipitis for ethanol tolerance. With what you found, would it be able to translate or rather transfer these genetic traits to other yeast species, for example, Saccharomyces cerevisiae, in order to improve its tolerance to ethanol, for example?
Yeah. That's a really good question. So I didn't really focus on the genetic basis why some strains had a higher ethanol resistance. It would have been really cool to see why. And then a major drawback of previously working with Stipidus was not being able to genetically modify it. And I'm not showing any of the work here, but a big part of my PhD was getting CRISPR to working it. So that is now a possibility, and it's fairly easy to create mutations and modify the genome. So it's definitely a possibility now, and it would be really cool to do. It's not something I will do, but if you wanted to talk more about it, I'm happy to.
Hi, thank you very much for your talk. So your work has focused mainly on the filamentous fungi from the Ascomycetes. According to you, filamentous fungi are more interesting than the Basidiomycete edible mushroom or not?
I wouldn't say they're more interesting. All fungi are cool. I'm not. That's a really good question. It's caught me off guard. The main reason why I chose to work with these species is that they are already used for food products. I'm not sure about the other ones you mentioned. And we're not limited to these. We are happy to try others. If you have any suggestions.
Yeah.
So yeah, I mean, we're happy to try as many as physically possible. Yeah, it's definite, like we're not closed.
Thank you.
Do we have any questions online? No. Okay. So we still have, I think, 4 minutes left. So one more question is okay.
This is just, I wonder like about the protein that you can produce. This is like easy for digestion for human.
That is something we're going to have to figure out.
Yeah.
To be honest, for example, corn and the mycoprotein that they produce, a lot of people are already eating it. You know, it's very it's on the market, and a lot of people are transitioning to that as opposed to animal-based meat. I am aware that some people are not able to digest it as much as others, but it's the same with all foods, I guess. No one's able to really digest things exactly the same. It would be something we would have to consider and test, obviously, because otherwise there's no point in producing something that not many people can digest. So yeah, it's a concern, but hopefully we're not doing something too dissimilar to what's already out there. We're just utilizing different wastes to grow the fungi on. So hopefully it won't be too much of an issue.
So we have 2 minutes. So one question is okay before we go to coffee break. Yeah, okay.
Hi, Chloe. I was wondering, are you going to start looking at the different wastes in combination? So you've got them separately at the moment, and then obviously yeast grown like YPD. Are you going to try and make your own version of that?
Yeah, I think we'd initially thought to do that if we couldn't get the yeast or the filamentous fungi to grow on one alone. As we've already seen that they can, is not so much of a concern, but it would be really interesting to see how if we combine different wastes, how that would affect the taste of the mycoprotein produced, the texture. And I guess that's a way of eliminating the processing downstream and having something really highly processed. If we can just grow it on a different feedstock and it will taste, I don't know, more fish-like if it's on the seaweed as opposed to the bread.
Nice.
Yeah.
That's really cool. Yeah. Thank you.
So I think now we are running out of time. Thank you, Dr. Chloe,
Thank you.
for your very excellent talk.